So hello, everyone. My name's Dr. Amanda Strayer-Scherer. I'm an associate professor and extension plant pathologist at Auburn University. I'm in the Department of Entomology and Plant Pathology. And as I'm an extension specialist, I'm also part of the Alabama Cooperative Extension's services. And today I was asked to give a talk on areolate mildew on cotton. Really, since I started in Auburn in 2019, this has become a pretty emerging disease, and I've been able to do a good bit of research on it, be part of some good collaborations. And so I'll give kind of an overview of the disease itself, and then get into some of the management practices and some of the research that we've been doing so far. So areolate mildew is also known as ramulary leaf spot, false mildew, or gray mildew, depending on what part of the world you're in. It's caused by two species of Ramulariopsis, Gossypii and pseudoglycines. Previously, it was also known as Ramularia areola and then Ramularia Gossypii and pseudoglycines, but Ramulariopsis is the current accepted genus for this pathogen. One of the good things about areolate mildew is that these two pathogens have a very narrow host range, and meaning that they primarily affect really just cultivated cotton species in the Gossypium genus. In terms of a bit of history, it's kind of fun to say as a plant pathologist that Ramulariopsis gossypii was actually first discovered here in Auburn in 1890. But it's been more recently detected in other parts of the southeastern cotton belt. We've also found recently pseudoglycines in Alabama, Georgia, Florida, Mississippi, and Tennessee as well. But worldwide distribution, it also causes pretty significant yield losses in Brazil, which they have both species. And so it's a bit of a challenge for them because they've had some pretty severe yield losses from areolate mildew. Gossypii is also present in India, and we also have several reports in Africa, including Nigeria, the Ivory Coast, which is in West Africa, Republic of Chad, Central African Republic, South Africa, and Tanzania and Uganda in East Africa. Now, one thing that's been kind of interesting, really about areolate mildew, even though it was discovered over a century ago, it really had a negligible impact on cotton production until really kind of recently. Disease has kind of intensified in Georgia and Alabama slightly to a lesser extent in recent years. And in 2018, disease incidence was actually high enough where we actually saw for the first time in over a century, this disease caused significant defoliation on susceptible cultivars, kind of in mid to late August in that year of both states. So a little bit about epidemiology. So the fact that, we really didn't see it for a while, at least, here in the United States is likely due to the fact that even though this disease is just labeled kind of as warm, wet weather disease, it really favors, that extended periods of humid and hot weather. And we've really seen it more in the subtropics and tropic regions, of the other parts of the world. Now, it really does require high humidity, more than 80%, kind of moderate temperatures, 60 or 68 to 86, and prolonged periods of leaf wetness. So some of you may be familiar with target spot, which is another disease on cotton. That one requires, a lot more frequent showers and heavy rain, where areolate mildew can really survive more on intermittent rain. And that's kind of where we see it kind of most active, periods where it's kind of intermittent rain, enough to get that prolonged period of leaf wetness, either due to dew. Irrigation can also increase your outbreaks. But we see it most often in the United States, kind of in July to August, where we have those frequent afternoon showers. And then, in India, they see it most prominently in monsoon season, kind of June to September. Now, in terms of how this pathogen volunteers, a little bit of the disease cycle. It can survive, from one cropping season to the next with, crop debris that are in the soil that have been infected, that had defoliated and fallen, rantoon cotton. So basically that means that for those of you hopefully familiar with cotton production, at the end of the harvest, when we get all the lint off the plants, they'll go back and kind of do some stalk destruction where they just kind of lop it off, kind of at, probably with about less than 6 inches of that cotton stalk still present, but regrowth can happen from that, and the pathogen can also survive on that. But also volunteer cotton plants. that show up kind of early in the season where we maybe didn't get a good enough crop destruction there. Now, going into the cotton season itself, conidia are then dispersed from one of those sources via rain, wind, and equipment, and then can germinate, within 12 hours once they reach, leaves, especially in that lower canopy. The fungal pathogen then enters through the leaf tissue with stomata, so those natural openings, via germ tube formation. Now, this is a polycyclic disease, so once you do get those infected leaves initially, when you have conducive conditions, so that, prolonged period of leaf wetness, high humidity, those moderate temperatures, and just activity going through the crop canopy, it will then move to infect other leaves within the plant canopy and move down the row or, side to side. So when talking about signs and symptoms of areolate mildew, another thing that I do appreciate about this disease is that it is very easy to identify. So typically we will see lesions appear first on the interior lower canopy, kind of at canopy closure. So about that first to third week of bloom. And, you know, even though I say we typically see it right in that lower canopy, areolate mildew will sometimes throw you for a curve, right? It'll appear on susceptible varieties like Delta Pine 2127 or Delta Pine 1646, kind of in that upper canopy first and then move downwards. But regardless of where it first shows up, those initial lesions are going to be what's kind of pictured here in figure A. The initial lesions will kind of be angular to irregularly shaped, approximately 3 to 4 millimeters wide. So we're talking very small lesions that will be yellow-green in color. Then as the disease progresses, you'll get this white powdery growth, which is pictured here in figure B. And essentially that growth is actual, white conidiophores and conidia that have developed after that leaf has been infected. And we typically see that on the underside of leaves. And then if the conditions remain conducive for the rest of the season, that pathogen will then spread, as I mentioned, throughout that plant canopy. And then those lesions will turn necrotic, and that's when you get the defoliation, which is pictured here in figure C. And so by defoliation, I mean those infected leaves will drop and fall, and we start to see yield losses there. And that's because, the primary source, right, of energy for plant growth is it's photosynthesis through the leaves, in addition to nutrients. And so basically, we'll start to get those yield losses because that plant can't get the proper energy it needs to, continue to produce bolls from those blooms. Now, kind of continuing on the topic of identification. Now, if you are a grower or a crop consultant that works with growers or another extension specialist yourself, really all you need is identification based on those signs and symptoms that I've had pictured in the previous slide. It's more than enough, right, to make informed management decisions. But if you're in, cotton research and you want to work on areolate mildew and Rabulariops, if you want to know what it is in terms of the species level for identification, so determining if it's Gossipii or pseudoglycines. I have a graduate student, Kimi Bariar, who started with a master's with me and is now a PhD student. So she's been working on this for a few years now. And we really struggled initially to get thorough identification to a species level of these pathogens. And what she's found is that, ITS alone is really not, so that internal transcribed spacer that's commonly used in fungal identification is really not sufficient on its own to differentiate between Gossipii and pseudoglycines for all isolates. So we started to take a multi-locus sequence analysis approach, or MLSA for short, where we found that really 5 genetic loci are required. So in addition to ITS, we also look at actin, the trans elongation factor 1 alpha, histone H3, and RNA polymerase 2, the second largest subunit, so that RPB2. And here's just kind of a short glimpse. Now, this is not published yet. She's currently working on finishing it up. We hope to get a publication out here soon. But you can see here kind of how these kind of group, but we do get differences between the isolates. Now, we don't know what those mean yet. That's another problem entirely, but really to identify if you have one of those two, you have to take it with this MLSA approach. So probably on to kind of my favorite part is, okay, we have this pathogen, so what do we do about it? Now, I will say that Brazil has been dealing with this a lot longer than we have, even though we first discovered it here in Alabama. as I mentioned, it really hasn't been a problem, right? And so in terms of multi-state variety and fungicide data for the US, a lot's not really available. We're trying to, right now, we've created an emerging pathogens working group in partnership with Cotton Incorporated to kind of help solve that issue. So we have about 9 to 10 states across the southeastern cotton belt currently participating in multi-state variety trials and fungicide trials to kind of get more collective data across the cotton belt. Because as those of you know who work in cotton, the performance of these varieties really can vary by location, and that's due to weather conditions, what your pest pressure is, not only disease but also insects and nematodes, your soil management practices, um, you know, pesticide inputs, what you have sprayed, irrigation practices, your seeding rates, plant spacing, which can all kind of help favor microclimates for diseases if you have them too close together, less flow through that canopy, and other factors. So currently, probably Alabama and Georgia, we probably have the most data in terms of varieties. So I just kind of wanted to present, excuse me, a snapshot, kind of some of our, variety data that we've collected since I've been here since 2019, at least in Alabama. And as you can see, we do have, some kind of good preliminary data. I would like to see how these varieties perform in our variety trials, across the Cotton Belt. But what we have seen is that Stoneville 4990, Delta Pine 1646, which has now kind of been phased out, Delta Pine 2127, Delta Pine 2012, and Phytogen 480 appear to be pretty susceptible to areolate mildew. And you can see some of that data from one of our trials at Field Crops Unit in Short, Alabama, which is just down the road from Auburn in central Alabama. And we also have some tolerant cultivars that appear pretty good going through the last several years of data in terms of tolerance. So I would not use the term resistance because they're still going to get disease, right? It's just an acceptable level of disease, which you can kind of see here with Phytogen 332 and 400 and Stoneville 5091. But in other trials, we've also seen Dynagrowth 3615, Delta Pine 2141, which is a nematode resistant variety, Delta Pine 2020, and Delta Pine 1840 appear to be more tolerant to areolate mildew. And I put, Delta Pine 1646 here, just a picture. And you can see that pretty severe defoliation if you look closely. So that browning there. And then the Phytogen 500 picture I posted up here. Phytogen 500 appears to be tolerant to areolate mildew, but one of the issues we've run into is that there's not one variety that will work well for both areolate mildew and target spot, which also occur in the cotton belt. And so Phytogen 500, even though it looks good against target spot or areolate mildew, excuse me, it's pretty susceptible to target spot. So hopefully down the road, we'll identify one that's good for both diseases because sometimes they'll occur at the same time and we want producers to have some good options. But they may just have to go with, what's been a problem in the past when making those decisions in the future. And another thing I wanted to highlight, I've been partnering with Henry Jordan, who runs the Auburn University Variety Testing Program. And so for the past few years, I've worked with him on this. And so these variety trials include crops in addition to cotton, but also, obviously, I'm going to highlight the cotton stuff. But, it also has data on peanuts, corn, sorghum, soybeans, small grains, and forages. And it's really an excellent resource for producers because you get unbiased data on these cultivar performances. And these locations are across Alabama, from South to North Alabama. And I've done disease ratings on trials in Central, Southeast, and Southwest Alabama for cotton, in addition to peanuts and small grains. And I mention that because these two are often good rotation partners for cotton. And so there is data available for producers or scouts on that. So if you scan this QR code here, or just simply type in Auburn University Variety Testing Program, it will take you to that selection platform. It's free to use. All you got to do is select, what crop you want to look at among this list. And then you can actually highlight certain research stations that are kind of closest to the conditions that you plant in. and see how these varieties perform, not only, to diseases, which is, my bread and butter, but also how they yield, how they grade, and it has a list of what production practices they utilized at those stations for the OVT trials. So when you're talking about making disease management decisions during the season, there's really limited options for areolate mildew. And honestly, fungicides are going to be your main way to curtail that, especially if you're between the 1st and 5th week of bloom. Typically, I don't recommend applications when you reach the 6th week of bloom, because you're getting close to defoliation. So if the disease shows up at the 5th or 6th week and you're close to defoliating anyway, it's just going to probably save you some money on defoliating, with those defoliants. And because it's already going to have some of those leaves drop, which you need to then harvest your cotton crop. So that being said, fungicides are not recommended for every field just because areolate mildew is there. You want to really think about what's in your 5-day forecast going out in terms of rain and temperature, what growth stage you're at when it shows up. And so with areolate mildew, I typically don't recommend, a preventative fungicide application, especially currently with this economic climate that we do have, and things are just expensive. And so in order to maintain those profit margins, I really recommend scouting. Typically, you're going to be looking for insects anyway. So just pull apart that lower canopy and look for signs of disease. You can apply it then and then do a second application if you need to. But typically, we've found that one application, is sufficient, which I'll show on the next slide, for most of the fungicides listed here. Now, even though target spot is not part of this presentation technically, right? It's on areolate mildew, but target spot occurs at the same time. And so I do want to give some tips on that because I think it can help when we're making decisions about areolate mildew. So, for that, we've recommended either preventative applications, based on growth stage, which you can do for areolate mildew, right? But typically, I see the most profit if you look more at a preventative application at the 3rd week of bloom, which I'll show in the next slide. But ultimately, I really recommend scouting, and you can do it based on the first sign of disease or the first sign of defoliation. Now, based on the research we've seen with target spot, I'm a little hesitant to say based on first sign of defoliation, because sometimes that disease can continue rapidly before you can get back out. And once we've seen strictly with target spot that once the defoliation reaches 25 to 30%, fungicides, it's very hard to recover that crop with a fungicide application. Now, that could be the case for areolate mildew. We don't have enough data yet to say that, but I would think it would be good to use that kind of as a cautionary tale, right? And do it more at the first sign of disease rather than the first sign of defoliation. Now, definitely if you reach 10% defoliation and you got rain in the forecast, I would apply a fungicide. Now, in terms of what options you have, I did include what's labeled for target spot because a lot of times that is present at the same time as areolate mildew, and you can see all these are labeled for target spot. Because this disease has really shown up more recently, the companies have kind of had to scramble to get this added to the label. And so right now we do have some special exemptions permission where you can apply these fungicides for areolate mildew. But they are labeled on cotton, but just caution there. They have been added at least to the label for TopGuard EQ, Revitec, Quadris, Priaxor, and Meravis Top. Now, one of the things I will caution with Quadris, especially, that's azoxystrobin, this is a very high-risk fungicide for fungicide resistance development. And based on talks with my counterpart in Georgia, Bob Camerite, he has personally seen some field efficacy failures with Quadris. And so what we're doing currently in my lab, as well as Alejandro Rojas' lab in Tifton, is we're looking to see if we can identify resistance to azoxystrobin, across the southeastern cotton belt. And my student Kimmy's also working on that. Now, into the heart of data, I would say, looking back, this is just one trial snapshot, again, of a trial we did a couple of years ago in 2023 at Field Crops Unit in Shorter, Alabama, that by far, compiling all my research, Revitec, if you're doing based on growth stage, just a single application at 12 fluid ounces at 3rd week of bloom has done very well. I would say I started looking at Quadris last year, which is going to be your more inexpensive option because there are a lot of generics of it, has done well, at least in our field trials in Alabama. But again, I would caution doing multiple applications of Quadris. And typically you would do it at 8 fluid ounces, which is not pictured here because I only have 1 year of data. So I'd like to get a little bit more data before I confirm or, deny if Quadris is really effective. But I would say also Priaxor, at 6 fluid ounces does well. And Maravis Top also at 13.6 fluid ounces, but really at the 3rd week of bloom. I would say it doesn't have as good of a, kind of, you know, curative activity where I've seen kind of with Revitec, even if you apply after you see signs of the disease, we still see some benefit from Revitec. So I think it's still able to knock it down a little bit. And it has a little bit better residual activity, at least visually from what I have seen. But Maravis Top, a lot of times if you apply it after the disease showed up, so in this case, this trial, we saw Areolate Mildew at the 4th week of bloom, so between these two applications. But at the 5th week of bloom, sometimes Maravis Top kind of falls apart depending on when that disease shows up. So again, that's why I really recommend scouting versus applying based on growth stage, at least at this time, for Areolate Mildew. Now, if a grower is to hold me to the fire, say, Amanda, I don't have time, to scout my plants, that's perfectly fine, right? But I hope they're still scouting, but they maybe can't get out enough. I would say, at the 3rd week of bloom is typically going to be your sweet spot most years, if I had to pick one. So the other things I kind of wanted to finish up with is just additional management considerations, just things that, they found that work in Brazil, and just some stuff we've seen anecdotally here in Alabama. So by far, a lot of this is going to be applicable to disease in any crop that you have. And the first one, of course, is going to be to reduce plant stress. So that I would say the biggest factor for that is nutrient management. So really consider soil testing to get your fertility levels prior to planting. And also maintain, of course, proper pH, and, using lime accordingly. But really the biggest one on here that I would say would be potassium for Areolate mildew. In addition to magnesium and calcium, those are also important too, because you don't want to be deficient in a lot of your micronutrients, and phosphorus as well. Now, with potassium in particular, potassium is associated with potash deficiency. And so basically, we start to see that show up right around the time that we see Areolate mildew and also target spot, right in kind of the peak week of blooms where the plants are starting to divert their energy from the leaves, from canopy growth to bloom formation and boll set. And so that means that when they're uptaking the phosphorus from the soil, they're diverting most of that phosphorus to what is the yield there, what the target is for us to harvest. And so you'll see kind of like a reddening of the leaves, just maybe sometime a wilting. But you'll also see a secondary pathogen show up, which is called Stemphylium. And that's not controlled with fungicides. It's mainly nutrition management or nutrient management, excuse me. And essentially, we see it a lot of times form a disease complex with Areolate mildew because it can also cause defoliation, where we see especially susceptible plants like Delta Pine 2127 or Phytogen 480 defoliate pretty rapidly with both of those present. So really managing potash deficiency is critical. And the other thing would be properly apply nitrogen and control plant growth. Now we have a bit more data on this regarding target spot, but when you look across crop systems, even in turf, we see excessive nitrogen fertilization or deficiencies in nitrogen can promote disease development. So in addition for cotton, poor canopy management, by not applying, plant growth regulators properly, and also, maybe over irrigating as well can get you rank vegetative growth, and it can create kind of like a humid microclimate within the plant canopy, which often favors disease development. And we've seen that clearly with our target spot research. It increases that humidity, increases that prolonged periods of leaf wetness because the canopy is so close together. And the taller it is, the more leaf tissue it has to interact with. And so based on what we learned with target spot, I think it's still a good approach for Areolate mildew. Now, probably one of the easiest things to do to prevent overwintering of that inoculum, and you may have already put this together based on my previous slides, is you want to rotate to a non-host. So it's One of the good things about airlate mildew, in addition to it being easy to identify, right, is that it only goes to cotton. So our other rotation partners like corn, peanuts, and soybeans are great for airlate mildew because that will reduce your inoculum going into the next season because they don't promote that pathogen growth as they're not a host. Now, one caution is if you do see target spot also present when you see areolate mildew, um, consider not rotating to soybean if you can, because that is a host for the target spot pathogen. Also, you want to eliminate crop debris where you can, so that's doing good cotton stalk destruction. And if you don't do, no till, at least do some reduced tillage practice to kind of, help degrade that inoculum source. Now, one great thing about this year, at least in Alabama and Georgia, is we've had a lot of rainfall. And that rainfall is actually going to increase the destruction and basically cause that crop debris to rot a little bit faster. So, in the case for this year in Alabama and Georgia, that's going to help reduce, hopefully, our inoculum carryover for a lot of these diseases, across all crops that can overwinter those pathogens. So that's really all that I have today. Here's my contact information. You're more than welcome if you have follow-up questions to email me, or here's my office phone. But I do want to really thank my lab mates. I cannot do this without the help of good people. Particularly want to thank Kathy Birch, who really, helps organize our cotton field trials. We do research, across 8 stations, for cotton, peanuts, and small grains. So it's a lot. So I also have Max Cavassa that does a lot of our peanut research. But a huge shout out to my graduate student, Kimmy. She's done really well with these projects and is going to be getting some publications out soon from her master's. So keep an eye out. And then, of course, Henry Jordan for his hard work and coordination of the OVT trials. Again, I think it's really an excellent resource for producers. And of course, Henry and I could not do all this without our station personnel across the state. And a lot of these projects that I highlighted and some additional ones that I didn't talk about were funded by Cotton Incorporated and the Alabama Cotton Commission. So again, I thank you. I know it's a lot of information in a short video, and so that's why I wanted to provide my contact info. And so that's all I have.